All right. Hello, everyone. Welcome to another session of our evolution seminar series. Today, we'll be learning all about infectious diseases as we're joined by Professor Jessica Hite from UW-Madison's Pathobiological Sciences. So without further ado, please join me in giving a warm welcome to Professor Hite.
Thanks so much. Thanks for joining me today for a conversation about the socio-ecological drivers of pathogen evolution in terms of virulence and drug resistance. So just a little bit about my background. I'm a quantitative ecologist, and in my lab, we use quantitative tools largely from the fields of ecology and evolution to understand pathogen evolution across space and time and from DNA to landscapes. So you're probably all very familiar that antimicrobial resistance is a growing public health challenge. Prior to the SARS-CoV-2 pandemic, the World Health Organization had named antimicrobial resistance as the #1 public health challenge. So globally, around 700,000 deaths are caused by drug-resistant infections each year. And these deaths are predicted to increase by 2050 to around 10 million deaths per year. So we really need to have this concerted effort to change the way that we're addressing drug-resistant infections. So just to put this in perspective, in 2019, deaths from these superbugs caused more deaths than those from HIV and AIDS. And this is largely affecting lower middle income countries like sub-Saharan Africa. But in an increasingly interconnected world, there are no boundaries to these drug-resistant pathogens or infections, right? And this is particularly true when we think about chronic infections. So many of you may know someone who's had a persistent drug-resistant infection, leading to long-term use of antimicrobials and recurrent visits to the hospital or the doctor's office to try and deal with this situation. And this leads to a substantial loss of quality of life. So major challenge here. And if we want to reduce this projection of 20 million deaths per year, or 10 million deaths per year by 2050, what is our current approach? So right now we put a large emphasis on monitoring, just where is drug resistance emerging across space and time. And we're often doing this in relation to not just a single drug, but multiple drugs, right? Because once bacteria evolve resistance to a single drug, it's very easy for them to evolve resistance to multiple drugs. So we can do surveillance methods to understand the emergence across time, and we're pretty good at this. And what we know from this monitoring is that if we increase drug usage, the prevalence of resistance goes up. And if we reduce drug usage, the prevalence of resistance goes down. And that's because this is a dynamical, evolving system, and bacteria have very rapid generation times, right? So we know that if we can get a handle on drug usage, we can drive this pattern down. That's one way to do it. But what we also know is that unfortunately, drug resistance continues to increase across time. And that is largely because despite repeated calls to reduce our over-reliance on antibiotics, we don't really have good alternatives at the moment. And it's very easy for us to overuse those in certain sectors, namely livestock. So we're going to talk about that more as we progress through the talk. So one reason we place such a large emphasis on monitoring is the idea is that if we can monitor and identify where we see emergence of drug resistance in the first place, then we can start to identify leaky pipes and reduce that emergence from even becoming a problem. And that's all well and good if we have very obvious point source pollution sites, like a large factory farm with runoff going directly into a waterway. Very difficult to address, but we can start to get a handle on it because we can identify a direct connection. Or say a factory that produces antibiotics. Largely, these are in India and China. We can reduce runoff pollution from those. But there are many other sources that we don't understand, right? We know that because drug-resistant bacteria don't acknowledge any boundaries, either across geographic boundaries or across species, then they're able to spread really rapidly through the environment. And while we're really good at saying, well, we see drug resistance hotspots in these particular areas, we don't really know what about the environment So the particular players in the environment, that means the plants, the animals, the hosts, the people, the climate. What about that environment is selecting for greater resistance and keeping it around? So both the emergence and persistence of drug resistance. We don't have a good handle on those types of questions. And that's what my research addresses. Because the reason we need a better handle on why it's emerging and what's causing it to persist is that we need to stop relying on just managing, right? So we've largely tried to just consist in this perpetual arms race where we create new drugs, bugs evolve resistance. We could keep doing this just perpetually, except that the cost to developing new antibiotics, both in terms of economic and time, are so substantial that big pharma has fled the field, and these same barriers prevent smaller startups from gaining any traction. So now we're left with trying to extend the lifespan, the shelf life of a dwindling number of effective antibiotics. We've done that in part by having stewardship management practices, right? So this is why you need a prescription to obtain antimicrobials in many parts of the world, but most of the world doesn't require a prescription for antibiotics. And so this is what's led to this increasing interest by the World Health Organization, because what they want to prevent is this emergency situation where we're thrown into containment and damage control of a rapidly spreading drug-resistant infection, much akin to something we were facing when we were dealing with the COVID pandemic, right? So we have this approach. How do we prevent this emergency situation? Part of that is that we need to do a better job at not just monitoring, but also improving our ability to accurately predict when, where, and why emergence is going to be highly prevalent and persistent, right? And so we're doing that in the lab using data-driven modeling and risk mapping. So today I'll tell you about a couple of projects where we're trying to use this more quantitative approach to understand what drives variation in drug resistance across space and time, right? Again, we're talking about the environmental players, all of the dynamics in that system, ecological players driving evolutionary outcomes. And so I'll tell you about that with a case study of E. coli across the state of Wisconsin, and then how we're trying to understand that spatio-temporal variation using some modeling approaches. Okay, so we've collaborated with the UW Health System and the School of Veterinary Medicine to collate data on the prevalence of resistance in E. coli across the state of Wisconsin. And we've mapped this. What you can see is that we have some emerging hotspots, not many, but a few, where we have resistance. Around 20% of those isolates are resistant to multiple different drugs. And what this map highlights is that there's quite a bit of variation. What's driving that variation, and how do we identify across this broad but limited geographic scale, how do we start to identify the leaky pipes where we could reduce that resistance from spreading even further and higher? So one obvious place to start in the state of Wisconsin is with livestock. And that's because in the US alone, where we have pretty good regulations on the use of antimicrobials in livestock, around 65 to 73% of all medically important antimicrobials are used in the livestock industry. These have historically been used as growth promoters because, for reasons we don't quite understand, animals grow much faster and with less food if they're given antimicrobials. Now, we put regulations in place back in 2018 to stop this practice, and many industries have kind of followed suit, except that they found a loophole. And instead of labeling these antimicrobials as growth promoters, they're now prophylactic treatment. So we still have quite a high usage of antimicrobials in livestock. The majority of all of our antimicrobials are used in livestock. And this pattern is much more prevalent in other parts of the world. Okay, so the reason we care about antimicrobial use in livestock is because, remember, we know that higher use of antibiotics and antimicrobials leads to higher drug resistance. We know that across multiple systems and with multiple drugs. And what we also know is that bacteria can move across hosts. So through the wonders of horizontal gene transfer, the genes that confer antimicrobial resistance are mobile, and they can move, say, from E. coli to E. coli, or E. coli to Salmonella. They can move from a Salmonella in the gut microbiome of a cow over to the gut microbiome of another cow, or they can move to the gut microbiome of a farm worker that's milking those cows, right? So we have mounting evidence that this cross-species transmission is happening. What we don't fully understand is if it's persisting. So just because transmission is happening doesn't necessarily mean that it's going to cause a problem, right? It could just be passed out of the microbiome very rapidly. Or there might be certain conditions where it's going to be more fully incorporated into the microbiome and lead to problems. So we want to get a better understanding of when, where, and why cross-species transmission is happening. So to do that, we have a retroactive study where we had samples, fecal samples collected from cattle, canine, and humans. And ideally what we would do would be metatranscriptomics, and we'd look at everything in these samples. But these are pilot data so that we can hopefully get more funding to go deeper with this type of question. So now all we've done is just DNA microarray analysis, and we focused on 16 genes that we know are important for drug resistance in E. coli. And what we find is that there's variation in the prevalence of resistance in E. coli across these isolates. The vast majority of resistance that we're seeing is occurring in dairy. Not surprising, right? They receive a large amount of antimicrobials. We know this. They're under high selective pressure for drug resistance to emerge. What we'd like to know is, in that smaller percentage of drug resistance that we're seeing in humans and dogs, is there genetic overlap indicative of cross-species transmission? And this is a really, really hard question to get at. So one way to do this is through statistical approaches. So we've taken these gene profiles and we've used a statistical approach that's similar to PCA. So except that it's non-linear dimensionality reduction. So all this does is takes these 16 genes and then collapses those down into two-dimensional space. And then we can map that and look at overlap in the genetic architecture of these different isolates across all of our species. And now we can split that up and look at how this genetic variation is playing out across our different hosts. And that's what this looks like. So it's a little bit confusing. Let's just hone in on a couple of different clusters, right? So first, let's just look at these bright orange groups. These are all of our susceptible isolates, right? So the ones that are still susceptible to our drugs. What we see here is that in certain regions, there is genetic overlap among these different isolates, right? But there's also this large swath in these susceptible isolates, say in humans, that have a very distinct genetic architecture relative to those in dogs and cows, right? So there's orange on this far right vertical side, and then there's gray in these other two groups, right? So there's no overlap there. So distinct isolates, no genetic mixing. But in these other areas that I've circled, there's a high degree of genetic overlap in these resistant isolates. That suggests that all of the isolates collected across these three different hosts share similar genetic architecture in specific genes. And this is indicative of cross-species transmission. The problem with this analysis and with most analyses to try and get at this question is that we can't quantitate it yet. So I can't tell you if this is I can't tell you for sure that it's not a 100% overlap. I can tell you that it's more of an 80% overlap, but it's really kind of a high, medium, and low level across these different genes. And that's really unsatisfying. We'd like to go deeper and do more quantitative analysis, which is what we're in the process of doing. So stay tuned for that. Okay, so first question is, do we see evidence that there's cross-species transmission happening? Answer is yes. The next question is, do we see variation across space? And the answer is yes. So Part of the variation across these maps is driven in part because unlike our large statewide data set where I showed you at the beginning, looking at overall resistance in E. coli across the entire state, these are haphazard samples. And so we don't have full coverage in every county. But what we can see is that in the counties where we do have overlap, say, for example, Wausau, there's variation in the percentage of resistance in all 3 different hosts. We know that this is going to be the case for any bug or any host or any state or country that we look at, right? There's just going to be a lot of variation in the prevalence of resistance. The bigger question is, how do we start to understand what's driving that variation in resistance, right? It's really, really hard to quantify transmission, right? As of the paper that you read, transmission is really, really difficult for us to quantify and parameterize unless we're directly measuring contact rates, say, across all farm workers, how many times they're milking a cow, and then we're sampling the farm worker and the cow repeatedly through time. It's really time consuming. It's really expensive. We don't have good data like that. So one way to start to get at this question, when you don't have the data that you need to address this, is through modeling approaches, right? So this is why statistical and mathematical epidemiological modeling is really powerful, because it can start to tell you where you might need to look across the environment and what type of data you need to collect to start to address that question. Okay, so if we are interested in what might drive variation across prevalence of resistance in the dairy system, one obvious place to start is different farm management practices. And one really big way that farms differ is in nutritional management. And they also have really good data on nutritional management because they're very interested in nutrition input, production output. So these kind of data are relatively easy to obtain. And the reason that they might be important for infectious disease and drug resistance is because, by and large, good nutritional management leads to healthier cows, leads to less disease, less drug use, less drug resistance. So that's one really obvious place to start there. If we're going to look in human systems, we might think about urban to rural gradient more and aquatic contamination, say in cities, and this could be an example of eutrophication. So we're doing that in other systems, but for now, we're going to talk about farm management and why that might matter. So to start to understand this question of how we address ecological and evolutionary feedbacks across these socio-ecological interconnected systems that are dynamical and multi-layered, we need to think about how pathogen evolution is occurring, right? So pathogens evolve in complex microbial communities. They're competing with all of these different microbes for resources, space, acquisition to tissue that they're going to then adhere to and infect the host. And so what this means is that if this microbial interactions are driving evolution, then ecological context either facilitates or inhibits the emergence of transmission and drug resistance, right? So in order for a drug-resistant pathogen to be successful and spread through the environment, it first has to transmit to a new host, right? And in order for it to transmit, it has to grow and reproduce enough to have enough offspring to make it to a new host. So this starts to lead to more of a mechanistic connection between the environment and evolution. And it suggests that resistance is an emergent property of the microbial community and the ecological context in which it is embedded. Now, if any of you study plants or wild animals, this all makes perfect sense, right? You would never study a prairie plant and think that its environment doesn't impact the dynamics of that plant population, right? And it's not that microbial ecologists and those who have studied antimicrobial resistance don't think that microbial interactions in an environmental context matter. It's just that we haven't largely approached this question from that perspective. For the most part, we've tended to study antimicrobial resistance of single pathogen isolates in a test tube or on a petri dish in a laboratory environment, and they're competing with other isolates for sure, but not other species. And we tend to investigate antimicrobial resistance under ideal growing conditions, right? Where everybody has access to unlimited resources. So we've largely removed them from this ecological context and the microbial communities, in part because we've had to, because these are very, very complicated questions. Okay, so can we use this framework of understanding microbial ecology and environmental context to start to get a better handle on what drives spatial and temporal variation in both the prevalence and persistence of drug resistance in different systems? And if we're going to focus on different types of farm management, where do we start? So we could say nutrition broadly, but nutrition involves many different components, right? So there's vaccines, there's probiotics, prebiotics, postbiotics, and phage, right? All of those are now being introduced as non-pharmacological alternatives to antimicrobials. And they're often roped in with whatever other management practices are happening. So we have this never-ending compendium of different management options on the farm. Where do we even begin to start to focus on this? Well, as a mathematical ecologist, you always begin by writing down equations and thinking about pathogen evolution and what drives fitness. And what drives fitness in this case, in terms of nutrition, is really just the pathogen's ability to grow and outcompete other microbes. So it doesn't really matter if their nutrition variation is coming from prebiotics or probiotics or postbiotics in a global sense. It just matters how that's affecting key parameters in the host system, so immune functioning. So how well is that prebiotic impacting the immune system, the ability of that host to clear the infection, and how is it impacting the growth of the pathogen? That's basically all we need to know in order to study transmission and pathogen evolution. So the beauty of these equations is that I can boil down these incredibly complicated dynamical systems into simple mathematical models, right? And then how does this work? So I'll give you kind of a primer and a caricature of how these dynamical models play out. All right, so if we're looking at resistance as an emergent property, and we want to understand microbial community interactions in an ecological context, where we're focusing on, say, just different nutritional management strategies, we put that into a set of ordinary differential equations, and we can keep track of the intake of food in a host. That's going to influence the gut microbiome, right? So all of these different microbes. And we're going to then look at multiple traits of these different microbes. So we can look at virulence, the harm that they cause to the host, their ability to transmit to new hosts, and then also resistance. And we can say that these traits are linked in different ways and look at how that plays out in this system. Okay, so now we have these resources that are supporting the gut microbiome, so these other beneficial microbes, but they're also being sequestered by pathogens for their own growth and development. And they're being used by the host to support a robust immune system. We know that immune systems are really costly to activate and support. And what we really want to understand is what's happening at the individual level in a way that scales up to affect the entire herd, right? That's because individual level pathogen production is what determines transmission to other individuals in the herd or the population level, right? So this may seem also very obvious, but what epidemiological models have tended to do is focus on either these within host dynamics or the between host dynamics. And what work in my lab does is link these two in a more mechanistic way. And because we can do that, we can start to look at feedback. So we can ask, how does higher infection levels in the herd scale back to influence recurrent infections and pathogen evolution in individuals? So now we have these cross-scale and bidirectional feedbacks that weren't previously accounted for in most of our epidemiological or evolutionary models. And they certainly weren't looking at nutrition, right? They're mostly assuming that there are these kind of phenomenological ways that pathogens grow in hosts, not mechanistically connected to different management practices. So the way we do this is using an adaptive dynamics approach. I apologize, some of these slides, there was a little bit of Mac PC hoopla that happened. So we use this an adaptive dynamics or a game theoretic approach. And all this is where we take a resident pathogen that has a given set of traits. It's infecting hosts in the environment. And we introduce a mutant. This mutant has a slightly different set of traits. It, say, requires a little bit more resources from the host to support costly traits like virulence. So it needs to cause more harm to the host, and to do that, it needs more resources. So we have these two different microbes, slightly different traits, and they're competing with each other. And what the adaptive dynamics allows us to do is run this system out over multiple generations, thousands of generations of these pathogens, and asks whether or not this mutant strain can outcompete the resident and spread through the population to the point that it becomes the new resident. And we do that by identifying an equilibrium, what's called the evolutionarily stable strategy. For all intents and purposes, this is a strategy that cannot be outcompeted by any other trait with any other pathogen with any other set of traits, right? So this is our new equilibrium level, the new dominant pathogen, right? We saw this kind of play out over and over with the SARS-CoV-2 pandemic, right? Where we had the Delta, we had Omicron, right? We had these different traits emerging as the dominant pathogen. Similar dynamics happening here, except in this case, so we're looking at traits like virulence and drug resistance. Okay, so what do our results look like? I'll show you, for example, one case where we have increasing levels of probiotic on the x-axis. On the top graph, I'll show you this evolutionarily stable virulence. So this is that winning strategy of virulence. So where is the virulence direction? What's the evolutionary trajectory of virulence under these different probiotic conditions? And then how does that affect the size of the epidemic at the herd level? And remember that virulence is harm caused to the host. And you may be wondering, well, we started out talking about drug resistance. Why are we now talking about virulence? The reason we're talking about virulence is because virulence is what drives drug usage on farms during an outbreak, right? So farmers aren't really thinking about drug resistance as they're managing their herds. What they're largely thinking about is how sick are my animals? Do I need to give them more drugs, right? And so if we're looking at how these different treatments are affecting virulence, that's going to give us an idea of which farms are going to be more likely to have outbreaks of more severe pathogens and increase their drug usage, leading to higher drug resistance. It also helps us understand what are the epidemiological and evolutionary consequences of all of these new non-pharmacological options that are coming on the market. Currently, our studies are very snapshot, and they don't really look at these bigger questions, these emergent properties of pathogens. And so that could lead us to a situation where we still have non-pharmacological options that are selecting for traits in pathogens that are harmful to the overall management of these populations, right? So there's these prebiotics and probiotics are changing the microbial community and the selective pressures in these hosts in a way that's going to also affect the evolutionary trajectory, and it can affect drug resistance as well. Okay, so these results suggest that at the top, we see that as we increase this probiotic, we end up selecting for higher virulence, but a lower or smaller epidemic. So what's driving these different outcomes? If we have this probiotic that's increasing immune function in this case, then the immune function is able to keep up with pathogen replication. It's a really robust immune system. And so the host is really rapidly able to clear the pathogen as that pathogen is growing and replicating. What this does is it ends up selecting for a pathogen that's going to speed up its growth, because it needs to grow faster to keep up with this immune system. And this can lead to a longer period of time, a more chronic infection where we have this longer window of shedding, where this host is clearing, but the pathogen is keeping up and it's spreading really fast because it's under this strong selective pressure. And so it ends up shedding, having to shed these pathogens, the pathogen propagules into the environment over a longer time frame. As this starts to kind of play out over time, the pathogen can't keep up with this immune function. And so then we can condense that down. And now we see that by condensing that down, this changes the selective pressures. This window now of when the pathogen is able to grow and reproduce and have this opportunity to shed into the environment to infect new hosts is condensed. So it starts using even more resources and becomes even more virulent to the host. So what this suggests overall is that if we use too much probiotic, we can end up in a situation where we've selected for more virulent pathogens. And this is bad for individuals, right? So if I have a super virulent strain of an infection, then I'm really sick. But because that window is constrained, the ability of me to have a prolonged infection and spread more propagules into the environment is also constrained. This reduces transmission overall across the herd or say to a classroom, and this leads to a smaller epidemic size. So what this suggests is that we really have to start thinking about both individual level and population level feedbacks to understand how different management options are going to drive pathogen evolution, both in terms of virulence and antibiotic resistance. So I've shown you this case where we've looked at this for virulence with this question of what the relationship is between drug use and virulence. So if we have more virulent pathogens in the environment, we're going to increase the likelihood that farmers are going to increase their use of drugs, leading to higher drug resistance. So this is just one example where we have a probiotic that greatly enhances immune functioning. And we've looked at this across different treatment interventions, and you have different outcomes depending on these two main parameters: how they influence pathogen growth dynamics, the shedding window, and that's driven largely by how they're impacting immune functioning. And so what these models help us do is identify data that need to be collected in order to parameterize, test the model predictions, refine them, and update them. So what many studies are doing when they're looking at both antimicrobial resistance, emergence in the environment, and non-pharmacological options to manage that drug resistance is looking at a couple of different components. So they might look at how a probiotic influences certain aspects of immune function, but not look at pathogen growth. Other studies will look at how this other product affects pathogen growth, but not immune functioning. So what we really need are studies that measure all of those traits in the same study so that we can start to have a more systems level understanding of these dynamics. And the final component that our models highlight is that we really need to understand the pathogen dynamics. So we need to understand transmission. And the best way to do that is to look at a time series of how transmission or propagules in the environment are playing out through time. So in the absence of data to address these broader questions, we can use quantitative tools from ecology and evolution to start to identify why we might see these patterns, and also what data we need to collect in order to gain a more mechanistic understanding of what's driving those patterns. Okay, so that's all I have for you today. I'd like to thank my lab, my collaborators and funding sources, and then say thank you, and we can open it up to questions.
Have people tested probiotics that pick out the pathogen?
They've looked at how probiotics will affect the microbial community overall. But getting at that question of when those different microbes are often beating pathogens. Anecdotally, yes, but mechanistically, those are still very preliminary analyses because the idea is that they've largely focused on competitive exclusion, but there's lots of different other dynamics that could play out that often aren't quantitated because of the way the studies are designed. Yeah, tend to be more, sort of taking a 16S approach and asking who's there and how are these different communities changing under these different treatment options. But that's largely kind of a one and done study as opposed to looking at what's happening through time and then quantitating those features. Yeah.
Just curious, in your modeling scenarios with the dynamics, you encountered scenarios where there is no ESS and instead you just have a limit cycle between highly and less virulent.
Yeah. So that's a great question. Yeah. So we have some modeling scenarios where what we call it is that these pathogens kind of ride the waves. So when there's low resources, they kind of go into this more dormant stage and hang out. And when you would normally think that they would go extinct, but they're able to just go into a tolerant phase and then emerge more robustly once resource levels start to occur. And so when we see that is when you have these non-equilibrial conditions. And by and large, those get missed because these studies are largely focused on equilibrial conditions. Yeah. And the only place that you see those non-equilibrial conditions leading to different outcomes is when you connect these within host and between host dynamics. So if you just focus on within host or you just focus on between host, you miss all of that dynamical feedback.
So you get that dynamic, you have a trade-off between within host fitness and non-host transmission.
Mm-hmm. Yeah.
I really hope we get funding to expand that Wisconsin E. coli study.
Yeah, me too.
It's really intriguing, but the spottiness of the data must be maddening.
Yeah, that one is really maddening. Yeah.
So, are those all types of strains of E. coli? Is the unit the species E. coli?
Across this one.
And so what's the logic of choosing that as the unit to look at?
It was mostly just because of just trying to use the most data we have at this point. So we have gone back and broken it up into different groups, but because of the sparseness of the data, we have, so that's just one of the projects. And we have another project where we did a deeper sampling. We have nasal swabs, we have both, but it's still really difficult to have uniform sampling, even just within the state. Yeah.
I was actually thinking going in the opposite direction, not partitioning by strain, but expanding the number of species you're looking at.
Oh yeah, to do.
Yeah, so if you looked at Salmonella and E. coli, would you find comparable distribution of resistance levels across the microbial species?
Yeah, that's a really good question. I'd love to do that too. We do have other projects looking at Salmonella. I guess. Yeah, hopefully we'll be able to expand that.
Pseudomonas or.
Yeah. Yeah, for sure. I think it was just because of this one, we had to do the plating and, right, so we do whole genome sequencing or transcriptomics, and we don't have to worry about that as much. Yeah. It's really good point. Yeah.
Do you have any sense at all, because obviously you weren't directly looking at it, but of how, so we were talking about, transmission between livestock and humans or, pets, things like that, how that may change in different places where like the local concentration of, like Wisconsin has a lot of dairy. If that is different in different places and if you have an expectation of how that might affect the dynamics of that transmission.
Yeah, for sure. We've been trying to get at that more with the spatial modeling, because it doesn't play out just with initial analyses. It doesn't play out in the way that you might think, right? So what we see in the human populations are really high concentrations in urban areas and less so in the more rural areas with high density. But part of that has to do right now, we think, with the samples that we have. And so we're trying to extend that to collect data from farm workers. But as you can imagine, a lot of those farm workers on dairy industry, they're all migrant workers. And so that's put a barrier to a challenge to that right now. So we have a project in place to sort of build those relationships. The other issue that's arising is that when we look at those genetic signatures, what the current data, limited as it is, suggests is that the overlap is higher at the county level than across counties. And so it's suggesting that there's some dynamical ecological interactions that are happening at the county level and not as much spread between counties, which is good if we're thinking about drug resistance, because part of what that map overlays is groundwater connections and looking at potential for connectivity through groundwater systems, through pollution from these high density farming areas. So what we're digging into a little bit more is to see if those high concentrations in urban environments, how much can we pull out how much they're driven by, say, living next to a lake in Milwaukee versus being connected to a watershed that's directly downstream from the farm. Really, really challenging to get at that question, but yeah. Chris.
There's a couple related questions in the chat from
okay.
Bill Saucier. He asks, what are immune enhancing prebiotics? And then I think this is related. What aspects of immune function are being measured to know that immune function is enhanced
Yeah.
by the prebiotics?
Yeah, that's a really great question. It's kind of your particular flavors of which 6 immune genes are you interested in looking at. I mean, a lot of these have focused on pro-inflammatory cytokines because what we know, for example, in salmonella, so a lot of the prebiotic, probiotic work, that's all done in salmonella systems, at least in my collaborations. And what we know is that inflammation really is great for salmonella growth and production and virulence factors. And so largely what the studies that we've been looking at are really focusing on are trying to tamp down inflammation, right? So that we can also then, reduce that cascade of cytokine storms that's going to have negative impacts on the host and help get pathogen growth under control.
And the other question is, what's the prebiotic?
So there's just, so if you do a search on prebiotics and probiotics for salmonella right now, you'll get like 3 million hits. There's just a never ending stream. So I'm not doing those tests exactly, and I don't have the test right off of my head. And also some of them are new products that aren't on the market and proprietary, and I couldn't mention them even if I knew all of them right now. So.
So I was gonna ask you, and we see a lot of MRSA and other drug resistant diseases, and especially things like C. diff, if one of our patients get them, no matter how careful we are, it tends to spread like wildfire throughout the unit. And I know that with C. diff, you can do like fecal transplants as a way of like getting rid of it essentially, since it's like an antibiotic resistant infection.
Yeah.
Are there like methods that are being studied similar to that are like non-drug related solutions to the antibiotic resistance?
Yeah, that's a really good question. I mean, those fecal transplants in C. diff is amazing, right? And so C. diff is another one we're trying to get funding to look at right now too, because one of the questions is whether C. diff is getting transmitted from dogs or cats.
It's more like you have one patient on the unit, and no matter how careful you are, it goes through the unit.
Yeah.
No other.
Yeah. So C. diff is really interesting. They've done really, I think, some of the best work on all of these different microbial interactions and competitive suppression, all the different variations of those competitive interactions in C. diff models, and looking at prebiotics and probiotics. So we have the best handle on that. So kind of all of this work is building off of the work in C. diff because it's so well developed. But as far as I know, fecal transplants aren't being used in livestock, but they're really exposed to a lot of feces. So I don't know that it's I don't know how you would, well, one, you could never, it's part of the problem, which is they're constantly exposed, right? But I don't know how you could quantify below, you know, you couldn't control, like you could give them a fecal transplant, but then they're also just gonna be constantly bombarded with all the other material in their pens. Yeah, that's a really good question then.
The DNA microarray that you showed earlier on, I think you said all 16 of the genes were functionally involved in resistance. Is that the case?
Yeah,
Might there be a role to play of convergent evolution in causing that signal?
Uh-huh.
Not trying to poke holes in your transmission conclusion, but if in those different contexts, the pressures are selecting for the same point mutations and amino acid changes, that those would cluster those genes together. And I assume the answer to this is that's why we want to get funding, so we can test this with neutral parts of the genome. But,
yeah.
you know,
Yeah, that's a great point. Yeah, absolutely. This was just a place to start to see if we even can see the signature of some sort of transmission, right? Because the null would be there's no, and they're all distinct and there's no overlap, right? So yeah, absolutely. Really great point. Hopefully you'll be on the review panel.
all right, well, we have no other questions from the general community. Thank you, Madam Fox.
How about you introduce myself on camera? Really, hi, Cecil is in there.
Oh,
Yeah,